herbal bacteria can absorb the light energy by the presence of a certain kind of chlorophyll and use the energy or the light energy in splitting H2O, H2S into hydrogen and sulfur. Hydrogen reduces carbon dioxide to form sugar and sulfur is precipitated. But that may take place in higher plants. But a group of scientists in California University in 1941 explained the mechanism of photosynthesis or the source of oxygen that releases as a result of photosynthesis by using the chlorella algae. Chlorella algae is supplied with the carbon dioxide containing the normal oxygen 16 and water containing radioactive oxygen 18. The products is the sugar, water, and the oxygen released as a result of photosynthesis, but the oxygen is the radioactive oxygen 18 similar to that in water. And also when the chlorella algae is supplied with the carbon dioxide containing radioactive oxygen 18 and water containing normal oxygen 16, the oxygen released as a result of photosynthesis is the oxygen 16 similar to that in water. So the experiment to prove that the water is the source of oxygen that released as a result of photosynthesis. So the group of scientists in California in 1941 proved by using the chlorella algae that the source of oxygen that released as a result of photosynthesis is the water, not is the carbon dioxide. But what about the steps or the mechanism of photosynthesis? Blackman explains that the photosynthesis process takes place by two successive steps. The first step is called light reaction. The second is called the dark reaction. What about the light reaction? In the diagram that appear on the screen, the sun is the source of light. The light can absorb it by the chlorophyll A and B. The electrons of the chlorophyll move from low energy level to higher one. So the chlorophyll become active chlorophyll containing great amount of energy. The active chlorophyll which containing great amount of energy use the energy in two manners or two steps. The amount of energy can use in splitting, or part of the energy is used in splitting H2O into hydrogen and oxygen. Hydrogen is carried on NADP to form NADPH2 and the oxygen is released. الووتر H2O لو عملنا له سبليتنج هيبقى هيدروجين واكسجين. لو سيبنا الووتر والاكسجين تاني هيحصل ريكومباينيشن ويعمل ووتر تاني. طبعا عشان ما يحصلش ده يبقى في كومباوند كمان اسمه ناد بي ياخد الهيدروجين ويعمل بيه ناد بي اتش 2 ويفضل الاكسجين لوحده فالاكسجين هنا ريليزد. يبقى انا عملت كومباوند اهو اسمه ناد بي اتش 2. The other part of the energy is used in change the ADP or adenosine diphosphate into ATP, which is called the adenosine triphosphate. So the two compounds that formed as a result of photosynthesis is the NADPH2 and the ATP. They move from the grana surface to the stroma. The light reaction takes place on the grana surface where the limited factor is the light. But the dark reaction takes place in stroma where the temperature is the limited factor of the dark reaction. So the dark reaction produces two compounds, NADPH2 and ATP. They move from the grana to the stroma and the dark reaction start to form it in the stroma. So the diagram of the light reaction is the sun, which is the source of light, absorbed by the chlorophyll A and B. The electron of the chlorophyll move from low energy level to higher one. So the light energy change into potential chemical energy. So the chlorophyll become active chlorophyll, containing a great amount of energy. Part of the energy is used in splitting H2O into hydrogen and oxygen. Hydrogen carried on NADP to form NADPH2, which is directed to the stroma where the dark reaction takes place. But the other part of the energy in the active chlorophyll change ADP into ATP. ATP also is moved towards the stroma where the dark reaction takes place. But what about the dark reaction? The dark reaction means reducing carbon dioxide by the hydrogen into sugar. So in the dark reaction, carbon dioxide is reduced by hydrogen that carried on net pH2 and the energy stored in ATP to form sugar. The dark reaction takes place in the stroma, but the light reaction takes place on the granular surface. The limited factor of the dark reaction is the light reaction is the light, but the limited factor of the dark reaction is the temperature. The experiment of Melvin Kelvin that proves the dark reaction that used the chlorella algae. By put the chlorella algae in the apparatus that appear in the figure. Put the chlorella algae in darkness place for about 12 hours for consuming all the food that's stored inside the chlorella algae. After that, supply the chlorella algae with the carbon dioxide containing radioactive oxygen, radioactive carbon-14. So the chlorella algae is supplied with the carbon dioxide CO2 containing radioactive carbon-14, and allow the chlorella algae to expose to light for 1 to 2 seconds. After that, put the chlorella algae in warm or hot alcohol to kill the protoplasm and stop the biochemical reaction. After that, we can carry out the analysis process of the compounds that formed as the result of photosynthesis in 1 second. Melvin Kelvin proved that the first stable compound that formed as a result of photosynthesis is the phosphoglyceraldehyde. The phosphoglyceraldehyde abbreviation is the PAGL, PGAL. The PGAL is the first stable compound that formed as a result of photosynthesis. Also, the Melvin Kelvin experiment proves that the photosynthesis process is not takes place in one step, but takes place in many steps, because the phosphoglyceraldehyde is formed of 3 carbon, but the sugar or the glucose is formed of the 6 carbon atoms. Also, the experiment of Melvin Kelvin proves the dark reaction. So this experiment of Melvin Kelvin proves the dark reaction, prove that the first stable compound in photosynthesis is phosphoglyceraldehyde, and also proves that the photosynthesis is not takes place in one step but in many steps. That is the diagram of the photo of the apparatus that used by the Melvin Kelvin. By use this flask which containing the chlorella algae, and that is a flash which act as a source of light, allows the chlorella algae to expose to light for 1 to 2 seconds. After that, put the chlorella algae in boiled alcohol that to kill the protoplasm and stop the biochemical reaction. Therefore, the experiment or the analysis of the compounds that formed as a result of photosynthesis in 1 second is PGAL. That's the experiment that proves the dark reaction. So, Blackman explains that the photosynthesis process takes place in 2 successive steps. The first is a light reaction, the second is a dark reaction. The light reaction takes place on the granular surface, but the light reaction takes place in stroma. The limited factor of the light reaction is the light, but the limited factor of the dark reaction is the temperature. The light reaction form net pH2 and the ATP as the compounds that formed as a result of the light reaction. But the dark reaction compound is phosphoglyceraldehyde in 1 to 2 seconds, but after that 2-BGAL or 2-phosphoglyceraldehyde change into glucose in the next steps of the light reaction. التلتين الاولانيين من شابتر 1، واحد بيتكلم عن الابزوربشن اوف ووتر اند سولتس، لما ابقى خلصتهم ابقى انا قلت تلت شابتر 1. الجزء الثاني او التلت الثاني بيتكلم عن الفوتوسينثيسيس بروسيس. نشوف شويه اسئله تشوف احنا الكونسبتس ديت اللي احنا عدت علينا، نشوف مفهومه كويس ولا لا، وبعد كده نبدا في التلت الثالث من شابتر 1. بنبدا كده بساينتفك تيرمز. Give scientific term for each of the following. The scientific study of food and various modes of nutrition of all living organisms. That is the concept of nutrition. So the answer is the nutrition. The next concept. Root cells have the impermeable superized ribbon called Casparian strip. That's not act as an important part. That's act as a enrichment information which is called the endodermal cells, which is a part of the endodermis. The next question. Vessels translocate water and salts from root to the leaf. The vessels means the tubes. Translocate means carrying or transfer water and salts from the root to the leaf. It is the xylem vessels. Xylem vessels has two main functions. The first is carrying water and salts from the root to the leaf and also support the plant. The next concept. The first stable organic compound which is produced in photosynthesis process. The first stable compound that formed as a result of photosynthesis process. It is the phosphoglyceraldehyde or PGAL, PGAL as a common abbreviation. The next concept. A reaction takes place on the granular surface to splitting H2O. What about the reaction that takes place on the granular surface and splitting it, where the splitting of H2O takes place? The reaction is a light reaction, which takes place on the granular surface, and the light is a limited factor. Limited factor means the reaction increase when the light increase and decrease when the light decrease until limited range or a certain range. The next concept. A molecule is the energy currency of all living cells or living organisms. The energy currency of the living organisms is the ATP molecule. ATP molecule means adenosine triphosphate, ATP. The next concept. An isotope that's used to prove the dark reaction. The isotope that uses the dark reaction is the radioactive carbon 14, because the normal carbon is 12. The next question. A scientist explains the mechanism of photosynthesis. The scientist that explains that the photosynthesis takes place in two successive steps, light reaction and dark reaction. The scientist is a black man, which explains that the photosynthesis takes place in two successive reactions. The first is the light reaction, but the second is the dark reaction. The next concept. A compound gives oxygen by its splitting up during photosynthesis. What about the compounds that split during the light reaction into oxygen and hydrogen? The compound is water or H2O. The next. The limited factor of the reaction that takes place in stroma. The question asks about the limited factor of the dark reaction. It is the temperature. Because the dark reaction acts as an enzymatic reaction, and the enzymes acts in a very low range of temperature. So because of the dark reaction is an enzymatic reaction, so the temperature is a limited factor of the dark reaction. The next concept. The passage of water molecule from high concentrated medium of water to low concentrated medium of water through semi-permeable membrane. في عندي 3 شروط اهم. high concentrated medium of water, وعندي low concentrated medium of water, وعندي semi-permeable membrane. يبقى احنا بنتكلم عن osmosis. So the osmosis is the passage of water molecules through the semi-permeable membrane from high concentrated medium of water to low concentrated medium of water through the plasma or the semi-permeable membrane. The next concept. The ability of colloidal substance to absorb water, swell, and increase in volume. The question asks about the colloidal substance. So that is the definition of the imbibition. Imbibition is the ability of colloidal substance to absorb water, swell, and increase in volume. The next. The passage of any substance through cell membrane against the concentration gradient by the help of some chemical energy released from the aerobic respiration. That's the definition or the concept of the active transport. The active transport is the passage of any substance through the cell membrane from low concentrated medium to high concentrated medium. That means against the concentration gradient that need chemical energy that produced as a result of the aerobic respiration. Aerobic respiration means oxidation of the sugar molecule at the presence of oxygen to release the energy that needed for the absorption of the ions against the concentration gradient. So the concept is the active transport. The next concept, a compound formed of 3 carbon act as a product of Calvin experiment. What about the product of Calvin experiment? What about the product of the experiment that proves the darker action? The compound is the phosphoglyceraldehyde or BGAL or BGAL. So that is the first stable compound that formed as a result of photosynthesis, or the compound formed as a result of photosynthesis in 1 second. الدياجرام ده هنشوف هيوضح لنا شوية اللايت رياكشن والدارك رياكشن علشان نفهمه أحسن. بقولك the following figure A and B indicate the result of these experiments were carried out on the culture of unicellular green algae to investigate the effect of the light and carbon dioxide for the formation of BGAL or phosphoglyceraldehyde. The experiment اتعملت على unicellular algae بيشوف تأثير الكربون ديوكسيد واللايت على الفوتوسينث بروسيس أو على الدارك رياكشن ازاي. نمبر A طبعا في الجراف الأولاني بدينا بكونستانت اماونت اوف بيجال، وبعدين حصل هنا لايت سويتش اوف عند النقطة دي. لما عملت لايت سويتش اوف هنا لقيت ان البيجال فورميشن increase until certain range and that still on a certain limit. لكن لما جينا في الدياجرام او الفيجر نمبر بي لقينا برضو نفس الليفل بتاع البيجال، لكن جينا عند البوينت دي وعملنا ريموفنج كربون دايوكسيد. هنا لقينا ان البيجال فورميشن ايه decrease. طيب هنسأل بقى، بيقول لك explain why bigal rises after the light is switched off in the figure A. ليه البيجال زاد هنا اهو لما عملت لايت سويتش اوف؟ البيجال لقيناه زاد كده، فبيسألني ليه لما اعمل لايت سويتش اوف البيجال فورميشن هنا زاد. نفتكر بس ان البيجال فورميشن act as a product of the dark reaction and the dark reaction limited factor is the temperature. So the source of light is the source of light and also temperature. So when the light switched off, the temperature become more suitable for the dark reaction. So the formation of bigal increase when the light switched off, but that takes place in until certain limit. After that, the level of the bigal stop increase. طيب يبقى البيجال هنا لما عملت لايت سويتش اوف البيجال زاد لان التمبريتشر هنا بقت مناسبه، لان البيجال فورميشن دارك رياكشن والتمبريتشر هي الليميتد فاكتور. Explain why bigal fall after carbon dioxide is removed in the figure B. طب ولما شلت الكربون دايوكسيد قال لي ليه؟ طب هو البيجال اصلا بيجي منين؟ From the reduction of carbon dioxide by the hydrogen carried on NADPH2 and energy stored in ATP. طيب انا ما عنديش كربون دايوكسيد، مين اللي هعمل له ريدكشن عشان يعمل بيجال؟ مفيش. يبقى البيجال يقل هنا ليه؟ because the bigal formed as a result of reducing carbon dioxide into bigal. طب يبقى لما اشيل الكربون دايوكسيد مفيش حاجه هعمل لها ريدكشن عشان تدي بيجال، يبقى علشان كده لما اعمل removing carbon dioxide the bigal formation decrease. Number C, what is the importance of bigal for the plant? مهم أوي البيجال ده للبلانت؟ مهم because the bigal act as a source of energy, produce amount of energy because it act as high energy compound. Number 2, bigal, from the bigal the sugar can change into starch and that form protein and the other compounds that formed in the plant. So the bigal is a source or the substance or the compounds that can change into starch, protein or glucose, starch and may change that into protein and fats. But in the diagram, what about if the light is switched off for a long time? لو انا عملت لايت، لما عملت لايت switched off البيجال زاد. طب لو فضلت عامل لايت switched off هيفضل زايد كده؟ لا، هيحصل له decrease. طب ليه يحصل له decrease؟ Because the formation of bigal needs the presence of net pH2 and the ATP that formed from the light reaction. يبقى من اللايت reaction انا بقى عندي net pH2 وعندي ATP، دول اللي بعمل بيهم reduction of carbon dioxide to form bigal. فما ينفعش يبقى في bigal وما فيش لايت. هو هنا انا عملت لايت switched off زاد البيجال لان كان في sufficient amount of net pH2 and the ATP that formed as a result of light reaction and move from the grana to the citron. برضو بنفس الميكانيزم، لو فضلت شايل الكربون دايوكسيد هيفضل يحمل decrease the formation of bigal until the formation of bigal is stopped. الجزء اللي احنا قلناه ده كان الجزء الاولاني من شابتر 1 اللي كلمني عن الفوتوسينث بروسيس، وكلمني كمان عن الابزوربشن اوف ووتر اند سولتس، وبعدين كلمني عن الفوتوسينث بروسيس. دلوقتي هندخل في التلت التالت من شابتر 1 اللي بيكلمني عن الدايجيشن اوف فود اور ذا هيتيروتروفيك نيوتريشن. What about the digestion? Digestion as a concept is a change or conversion of large food molecules into small food molecules at the presence of catalyst as an enzyme. يعني انا عندي البروتين بيج موليكيول عايز احوله لامينو اسيد، يبقى انا احول البروتين لامينو اسيدز، ابقى انا لازم اعمل digestion. digestion ده need the presence of enzymes، وهنقول بيعمل ايه. يبقى نرجع هنا تاني، يقول لي digestion is the conversion of large food molecule or polymers as a protein, starch and fats into smaller ones or monomers as the amino acids and the glucose. الدياجرام اللي قدامنا بيقول لنا كأن الانزيمز دي مقص وبتقص لي اليونتس بتاعه البوليمرز عشان تحولها لي لمونومرز. طب وانا بحول البوليمرز للمونومرز ليه؟ to allow the monomers or to help the monomers to pass through the blood capillaries to reach the blood, which transfers the digested food to the whole body organs. This can't allow the big molecules to pass through it to reach the body cells. But the monomers can transfer to the blood through the absorption process and can transport to the whole body cells, which act as a source of energy and help in growth and repair the burned out tissue. But the digestion process takes place at the presence of a certain catalyst which is called the enzyme. Catalyst يعني عامل مساعد بيسرع الرياكشن. احنا نعرف ان الكاتاليست ده accelerates the rate of the reaction still as it at the end of the reaction and can't enter the reaction. طب لما اجي اشوف what's meant by the enzymes. Enzymes are proteinic substance have the properties of catalysts. The enzyme had the specific ability to activate particular chemical reaction. يعني الانزيم اللي بيشتغل على البروتين ما بيشتغلش على الفاتس، بيشتغل على الفاتس ما بيشتغلش على الستارش and so on. يبقى لما اجي اشوف الكونسبت of the enzyme, it is a proteinic substance has the properties of catalyst and has specific ability to activate particular chemical reaction. What about the types of mechanisms of enzymes? هي الانزيمز تعمل حاجه من اتنين، يا بتعمل لنا breaking down يا بتعمل لنا building up. يعني ايه breaking down؟ يعني بتكسر لي البوليمرز لحاجات اصغر منها بسميها مونومرز. طب ويعني ايه building up؟ يعني اجيب المونومرز اجمعها اعمل بيها بوليمرز، وكنا بنسمي الحكايه دي polymerization process. يبقى الانزيم ممكن يعمل breaking down، ممكن يعمل building up، لكن كمان في انزيمز have reversible effect. في انزيم ممكن يعمل breaking down ويعمل building up حسب حاجه كده يمكن تكون عدت عليكم اسمها لو شاتيليه. يعني قاعده كده الرياكشن ممكن يمشي في الاتجاه ده، ايه الشروط بتاع انه يمشي في الاتجاه ده وايه الشروط اللي يمشي في اتجاه الفورميشن اوف ذا رياكتنس لغايه ما يتساووا الاثنين. هنشوف دلوقتي what about the properties of enzymes. الانزيمز they are specific in their functions، متخصصة قوي. Enzymes accelerate the rate of the reaction until it reaches the case of equilibrium. يفضل الرياكشن دوت يزيد لغاية ما يوصل للإكوليبريم. The enzymes is still as it at the end of the reaction. Enzymes can be used over and over again. Some enzymes may have reversible effect. Some enzymes are secreted in inactive state, and the enzymes are affected by the temperature and the pH. أنا لما أجي أقول البروبرتيز أوف enzymes ديت هلاقيها كتيرة. طب أنا أفتكر هو الإنزيم ده مش كان كاتاليست؟ في الديفينيشن بتاعه بنقول بروتينيك سبستانس has a properties of catalyst. طب أجي أشوف الكاتاليست إيه؟ إحنا قلنا إن الإنزيمز بروتينيك سبستانس، وقلنا إن عندها specific action. والكاتاليست ده بيعمل إيه؟ accelerate the rate of the reaction. طب وبعد ما يعمل accelerate the rate of the reaction يجرى له إيه؟ still as it at the end of the reaction. طب ولما يفضل زي ما هو بعد الرياكشن اعمل بيه ايه؟ استخدمه تاني، يبقى the enzyme can be used over and over again. وبعد كده اعرف ان في حاجات بتاثر على الانزيمز. the enzyme affected by temperature and pH. نلاقي some enzymes are secreted in inactive state. يعني ايه inactive state؟ يعني ما بيشتغلش. طب والانزيم لما بيشتغلش يبقى احنا بنطلعه ليه؟ السيلز بتاعتنا made of protein. يبقى اي انزيم هيعمل دايجيشن للبروتين لو هو اكتف هيهدم اول حاجه السيل اللي عملته. يبقى هنا لازم يبقى ان اكتف، وبعدين يروح كافيتي تيجي حاجه تانية تعمل له اكتيفيشن. يبقى some enzymes are secreted in an active state to protect the cells which secrete the enzyme from the action of the enzyme. وبعد كده some enzymes have reversible effect. يعني في انزيم يعمل breaking down building up at the same time. دي احنا بنقول عليها properties of the enzymes. قدامنا هنا الاكشن بتاع الانزيم. لما يبقى عندي ادي انزيم ويجي كومباوند بالشكل ده بوليمر، يجي الانزيم يدخل يقسمه لاتنين مونومرز بالشكل ده. بعد ما كان عندي كومباوند مثلا الكبير اللي قدامنا اللي لونه جرين ده هيتقسم لتو كومباوندز اصغر منه شويه، بنسمي دول مونومرز او برودكت A and B. لما نيجي نشوف ال digestive system of the human beings. The digestive system of the human beings start with the mouth opening. The mouth opening is the beginning of the alimentary canal. The alimentary canal consists of the mouth opening, which leads to the buccal cavity, leads to the pharynx. The pharynx leads to the esophagus. The esophagus carries the food to the stomach. The stomach leads to the first part of the small intestine, which is called the duodenum, and the rest of the small intestine at the ileum. The small intestine is connected with the large intestine. But the accessory glands of the digestive system are the salivary glands, liver, and pancreas. السلايفري جلاندز دول مهمين جدا. الكانال او التيوب اللي بيمشي فيها الفود مره تزيد قوي وتبقى لارج، ومره تقل قوي وتبقى سمول، ومره تعمل ساك نسميها ستومك. كل ده بنسميها دي alimentary canal. هنبدا نشوف اول حاجه في الدايجستيف سيستم the mouth. The mouth containing teeth which are differentiated into incisors for cutting the food, canines for tearing the food, premolars and molars for crushing and grinding the food. الانسيزرز الاولانيين دول اللي قدامنا دول 4 اهم، لكن من هنا دي واللي جنبها من الناحيه التانيه دول بنسميهم كاناينز. وبعد كده الدروس اللي بنقول على دول بريمولارز، وبعدين المولارز دول. ده الستراكشر او التايبس او different types of the teeth. Inside the mouth is the presence of the tongue. The tongue has many functions. The first is the manipulation of the food. Manipulation as the lubrication of the food or mixing the food with saliva. Tasting the food. Help in talking. Help in swallowing the food. The mouth also containing 3 pairs of salivary glands, and they secrete the saliva. What about the saliva? The saliva containing mucus which lubricates the food to facilitate its swallowing. and also containing enzyme which is called the ptyalin enzyme. Ptyalin enzyme or salivary amylase. That is the enzyme that can hydrolyze starch into maltose. Starch is a polysaccharide, but maltose is a disaccharide. So the first compound that formed as a digestion inside the human digestive system is the maltose. The maltose is the first product that formed as a digestion in the human beings because the starch changed into maltose by the presence of ptyalin enzyme. The ptyalin enzyme acts in the weak alkaline pH as 8 pH. pH means the potential concentrated hydrogen ion or the power of hydrogen. That's the first part of the digestion or where the digestion takes place. What about the pharynx? The pharynx is a cavity at the back of the buccal cavity or the mouth. It leads to both trachea and the esophagus. اللي قدامنا دي التراكيا اهي، والاسوفيجاس اللي جنبها الناحية التانية. الجزء اللي فوق ده احنا بنسميه pharynx. pharynx ده هنا مهم أوي لما اجي اعمل swallowing. تلاحظ ان التراكيا دي raises upwards to block the glottis or the epiglottis block the glottis. يبقى الفتحة بتاعت التراكيا هنا بتتقفل، الفود ما يلاقيش مكان غير انه يعدي للإسوفيجاس، واحنا بنسمي دون دي reflex action. هنيجي هنا نشوف الفيديو دون هيوريني ازاي بيحصل digestion in the buccal cavity. طبعا دي piece of food احنا بنسميها bolus. الفود ده بيحصل لها digestion او manipulation inside the mouth. هتبدأ بقى الفود ده هيعدي هنا، هناخد بالنا وهو بيعدي هنا ان الابيجلوتس دي بلوك ذا جلوتس، هتقفل الجلوتس دي. يبقى هنا الفود هيعدي للإسوفيجاس، واحنا قلنا ده act as a regulatory reflex action. اهو الفود عدى الجزء ده اهو. لما نيجي نشوف الفود لما يجي يعدي هيعدي يروح للإسوفيجاس ده. The food move from the pharynx to the stomach through the esophagus. The esophagus is 25 centimeter tube that connect between the pharynx and the stomach. It lies between the two lungs or pass between the two lungs parallel to the vertebral column. It runs through the chest cavity and penetrates the diaphragm until reach the stomach. The food passes through the esophagus by peristalsis. Peristalsis is a series of contraction and relaxation. يبقى اعمل contraction كده فوق البولس وريلاكسيشن تحتها، فالبولس تنزل تحت، اعمل contraction تاني فوقها واعمل relaxation تحتها، يبقى كده اسمه series of contraction and relaxation. ده اللي احنا بنسميها peristalsis which allow the food to pass through the digestive system. الفود كده وصل لغاية الستومك. نيجي نشوف ايه اللي بيحصل في الستومك. طبعا الجزء ده بيوريني شكل الجلوتس والإبيجلوتس وازاي انها بتتقفل. برضو هنا ادي الفود ما ينفعش يدخل هنا التراكيا، وبالتالي تيجي الإبيجلوتس دي تقفل الجلوتس علشان الفود يعدي يروح لمين؟ يعدي للإسوفيجس. نفس الكلام بنشوف شكل التراكية الأبر بارت اوف ذا لارynx اللي فيها ادي الجلوتس ودي الإبيجلوتس اللي لما تطلع التراكية لفوق دي تسدها، وبالتالي الفود ما ينفعش يعدي في التراكية وبيعدي للإسوفيجس. الشكل اللي قدامي ده بيوريني البريستاليسيس. يعني انا عندي لو عايز فود يعدي يبقى اعمل كونتراكشن فوق ريلاكسيشن تحت. ده اللي احنا بنسميها the peristalsis process. Inside the esophagus, the presence of glands that secrete mucus only, and they can't secrete digestive enzymes. So no digestion takes place in the esophagus, but the esophagus containing mucus just to facilitate the passage of the bolus through the esophagus and to reach the stomach. the circular muscles along the esophagus wall that is in the alimentary canal undergo a series of contraction and relaxation known as the peristalsis. What about the stomach? The stomach is a dilated muscular sac at the left side of the abdominal cavity, just below the diaphragm. يعني لما اجي اشوف left side of the abdominal cavity، مجرد ما ابقى احط ايدي كده تحت الديافرام بالظبط، ده مكان stomach. قصاد الاستومك هنا liver، تحت الاستومك هنا في بنكرياس، جنبه كمان شويه هلاقي سبلين. ده الاستراكشر بتاعنا او السايت اوف ذا ستومك اند ذا ليفر. يعني ما ينفعش حد يقول لك ايه انا معدتي وجعاني، لا معدتي وجعاني تبقى مش هنا، يبقى احنا بنشاور على الجنب اللي تحت القلب او تحت الديافرام، هنا هو تبقى كده بنتكلم عن ستومك. طيب stomach containing or start with the sphincter that's called the cardiac sphincter. cardiac كل ما بنقولها يعني حاجة related to the heart، حاجة تبع القلب، عشان ده السفنكتر اللي جنب القلب. The cardiac sphincter allow the passage of the food to pass from the esophagus to the stomach and preventing its return back. And also the stomach end with the pyloric sphincter, which act as a muscular sphincter that allow the food to pass from the stomach to the duodenum and also prevent the substance to flow back. What about the properties of the gastric juice? Gastric juice means the juice of the stomach. The gastric juice is colorless. It is liquid containing about 90% of water, and the rest or the 10% is the HCl and pepsinogen. So what about the function of the HCl? HCl has 3 functions, and the pepsinogen also very important in active enzyme, but when the pepsin or pepsinogen become active pepsin that can digest the protein. الفيديو ده بيوريني شكل البولس، البلعة يعني اللي بتعدي في الكارديك سفنكتر بتوصل لغاية الاستومك. هنا تبدأ الاستومك تعمل سكريشن للجاستريك جوس. الجاستريك جوس اللي موجودة هنا 90% water لكن 10% HCl and pepsinogen. The medium inside the stomach is 1.5 to 2.5, which act as a strong acidic medium. What about the function of HCl? It makes the stomach medium acidic 1.5 to 2.5 to activate the gastric enzyme, so that the action of thiolin enzyme stop because it acts in alkaline medium. التايلين اللي سميناه salivary amylase بيشتغل في الماوس في ويك alkaline medium 8. طب لما ينزل للستومك strong acidic 1.5 يبقى هنا التايلين مش هيشتغل، لأن التايلين act in weak alkaline medium. And we explain that the enzymes are affected by temperature and pH. So the change of the pH value from 8 to 1.5 that allows the thiolin enzyme to stop its action. So the HCl responsible for makes the medium strong acidic 1.5 to 2.5. That's very important to activate the pepsinogen into a pepsin, and also very important to kill the harmful living organisms which may get with the food in the human stomach. But what about the active pepsin? The pepsinogen is an active enzyme because the active enzyme digests the protein. The cells are made of protein, so pepsinogen is active to protect the cells against the action of the enzymes. So the pepsin, the active enzyme, can hydrolyze protein into polypeptide. The pH value of the stomach is the 1.5 to 2.5. احنا علشان ما نتعبش كتير في البي اتش احنا نقدر نعرف ان الاليمنتري كانال كلها weak alkaline medium except the stomach strong acidic medium. الاسيديتي بتاعت الستومك دي كان عاملها ال HCl، فال HCl مهم عشان يعمل لي الاسيديك ميديوم، عشان ده اللي هيأكتيف البيبسينوجين لبيبسين، وده الميديوم اللي البيبسين بيشتغل فيه فعلا عشان يعمل لي hydrolyzing the protein into polypeptide. دي كانت الفانكشن بتاع ال HCl والفانكشن بتاع البيبسينوجين اللي احنا قلنا بيعمل هيدرولايز بروتين انتو بوليبيبتايد. احنا لغايه دلوقتي قلنا 2 انزيمز بس. قلنا تايلين in the buccal cavity وسميناه salivary amylase, hydrolyzed starch which act as a polysaccharide into maltose which act as a disaccharide. ستومك ما عندهاش غير انزيم واحد بتشتغل على فود سبستانس واحده، يبقى الانزيم ده اسمه بيبسين hydrolyzed بروتين into polypeptide. طب الستارش بيتاثر في الاستومك؟ لا ملوش دعوه. طب الفاتس بتجرى لها حاجه في الاستومك؟ ما بتجرى لهاش حاجه. just amount of protein change into polypeptide due to the action of the pepsin enzyme. What about the give reason that the stomach is not digest itself, or why doesn't the stomach digest itself? Because the stomach secretes mucus layer to protect it against the effect of the juice. Pepsinogen doesn't become active until it reaches the stomach cavity. Number 3, HCl is secreted as a tip of the cell. يعني باختصار الاستومك مش بتهدم نفسها لأن الاستومك thickened internally with a thick layer of mucus. الميوكس ده هيمنع الجاستريك جوس انها توصل للسيلز. في نفس الوقت البيبسينوجين ان اكتيف، هو مش هيبقى اكتيف الا لما يجي على الفود في وسط الكافيتي. ال HCl بيطلع من التبس اوف ذا سيل، من الطرف بتاع السيلز. كمان ده بيبسينوجين and HCl each of them is secreted from side away from each other. يعني المكان اللي بيطلع لي بيبسينوجين بيختلف عن المكان او بيبعد عن المكان اللي بيطلع لي HCl. كمان HCl is secreted in diluted state until reach the stomach cavity where the hydration takes place to absorb water that increase the pH or increase the acidity of the HCl. طيب يبقى كمان عرفنا الفانكشن بتاع ال HCl، الفانكشن بتاع البيبسين، وعرفنا why the stomach is not digest itself. What about the next part of the stomach? It is the small intestine. The first 25 centimeter of the small intestine is called duodenum, but the rest is called the ileum. What about the duodenum? The duodenum is a part of the small intestine which receive the pancreatic juice and bile juice, and also that act as a part of the small intestine which containing a part of the intestinal juice. So the duodenum, the first 25 centimeter of the small intestine, receives the pancreatic, the liver secretion which is called the bile juice through one common duct called the hepatopancreatic duct. The ileum, it is the rest of the small intestine where digestion is completed and the digested food is absorbed. طيب نرجع كده تاني. لما اجي اشوف الديودينم ايه الجيوسس اللي بياخدها. كان البايل جوس which is secreted by the liver. The bile change fats into emulsified fats. It's secreted by the liver but stored inside the gallbladder. يعني محدش يقول لي ان الجول بلادر responsible for the secretion of the bile juice, but the bile juice is secreted by the liver and stored inside the gall bladder. The bile change fats into emulsified fats by decrease the fats into small pieces to increase the areas that exposed to the enzyme. But the enzyme which secretes which digests the fats is a lipase enzyme which is secreted from the pancreas, change the fats into or emulsify the fats into fatty acids and glycerol. But also the pancreatic juice containing NaHCO3 or sodium bicarbonate. The sodium bicarbonate is the strong alkaline medium or strong alkaline substance that neutralize the acidic medium and make the medium weak alkaline. الحقيقة الوقت جري بسرعة، ما لحقناش نكمل باقي الشابتر، لكن ان شاء الله الحلقة الجاية بإذن الله نكمل الشابتر بتاعنا ده. أصدقائي الأعزاء، شكرا لكم وإلى اللقاء.